So we continue with the next session on using prior knowledge in process development and manufacturing strategy. And I leave the floor to Cauwenberghs from the Irish agency.
Thanks, Mats. So this session is going to cover prior knowledge in process development and manufacturing strategy. We've touched on it a little bit already in this morning's session, but we're going to focus more deeply now on how, or what is the scope for prior knowledge during development, and there is indeed quite a lot of scope for it to be used. There's a lot of areas where prior knowledge could be used in development. I'm not going to go into them all. I've decided to focus on some few areas, but we can use it in risk assessments in terms of assigning criticality of process parameters, possibly in justifying the ranges of the PARs, and maybe in justifying the emission of some of the product-specific studies, for example, certain resin lifetime studies, which I won't go into in my talk, but we'll see in some of the case studies later on. So there's a lot of open questions, and we've heard some of them already this morning, and hopefully throughout this workshop we can answer some of those questions and come to some agreements. So how can the prior knowledge be integrated into the process development and control? And this is something I'll come back to in terms of integrating prior knowledge, as opposed to it be something separate that you just refer to, but how do you integrate it into each product? And how do you balance, you have your product specific data and prior knowledge, and how is that balanced in the dossier? And then how could this be presented in the dossier? And as regulators, I guess we need to strike a balance between the need for product specific data and regulatory flexibility where appropriate. And we have obviously some models and solutions that we use already today in terms of post-approval validation, small-scale models, post-approval change management protocol. But we haven't really, I think, seen the full potential of prior knowledge yet. And I bring up this idea of a holistic approach where prior knowledge is integrated into all these other tools that we have in terms of process development. And at least as I see it, there are a lot of areas in process development where you can get prior knowledge from, as we've heard about already. There's scientific knowledge, there's knowledge from vendors in terms of container closure systems, filters and so on. There are the platform processes, which you've heard about this morning. What I'll focus on a bit more, risk assessments and small scale models. And of course, then obviously in each submission of your product specific data. But so there is a lot of scope to leverage these data for process specific developments. So as regulators, we accept that there is a huge amount of knowledge that is gained through the development of multiple, I've used the word here, Keith, you won't like this word, similar, you're right, you used a better word because we want to avoid the confusion of similar or biosimilarity or anything like that, but similar, and it's a tricky question. How do you show that? That's the crux of the argument. How do you show that the other products are relevant for yours? And that's not something we can probably completely address today. It'll be something that'll evolve over time. But at least if the manufacturing process has the same or closely similar manufacturing steps, uses the same type of equipment, and manufactures a similar, in inverted commas, product, then data from the control of other products could be relevant. And as regulators, we're happy for that data to be submitted with appropriate justification, and the justification is an important part of that. So just focusing on some areas where there is scope and possibly we could get agreement on for prior knowledge. So we've heard already about some of the risk assessment approaches, and these are very common now in many dossiers. So I've just taken an example. If we just take a theoretical example of a chromatography step, it doesn't really matter what it is. So you've a standard risk assessment approach, maybe an FMEA or so on. And what you'll get, so these would be process parameters from chromatography. You might get some risk prioritization numbers where your risk assessment says some things could be carry more risk than others. And then for those that are over a certain cutoff, you may do some process characterization studies to further study those. And the ones that are lower risk, you may say they're non-CPPs, and we will do no further in-depth study on those. And on the basis of that process characterization, then you might decide some are critical and non-critical because you have additional, maybe small-scale data to justify that. So could it be considered that based on a platform or considerable data from previous products, that the low-risk process parameters from a previous one can just be considered low risk for the next one? I think possibly if the data is solid to show that the other products are representative, and that if you run a certain manufacturing step in the same way with the previous number of products, and we run it again in the same way for our next product, it's likely that what was low risk is still low risk. It's not suddenly going to become critical. I've picked a sort of maybe a controversial example here of a way this could be done in terms of aggregates. You could, rather than just cross-refer even to your FME or your risk assessment, you could tweak it along the way. If you think of severity, occurrence, and detection, you might say, severity might be the same, or it might be different. It depends on the patient population. If how severe, say, aggregates might be. Would it occur to the same extent? Depends on, could be product-specific factors. Could it be detected in the same way? Would you use the same analytical method? So this is just a theoretical example of how you could use a previous risk assessment, but also update your risk assessment for certain bits as you go along. And so if we look at it almost in a decision tree type approach for process parameters, you have all your process parameters, and then can prior knowledge be used to stratify those initially into low risk and the ones that are used for further study. I think that's possible. And then moving on from those ones that you would normally further study, is there a role for prior knowledge to, stratify those again into your final list of CPPs and non-CPPs? And feel that studying the same low-risk process parameters over and over again in repeat submission for similar products may not be necessary in some cases. And it could allow us as industry and regulators to focus on the process parameters that are really the ones that are critical in quality and being shown, say, across a platform, that are the ones that could really affect the criticality of the product. And in terms of, for those critical ones, even moving on from that, what you might study in the models, for each manufacturing step, there's a whole plethora of quality attributes you might want to study to justify that those models will show you what's the true case in the full scale. But if you've done this many, many times, or several times at least, you can show that it may be the case that the same process parameters always impact the same quality attributes. As I talked about earlier, you have inputs and outputs. And if you have your platform or your similar products, and you can show us that these process parameters always impact these quality attributes, so these are the ones that makes most sense to justify to study in the small scale models. That's a way of, I guess, deciding up front what attributes you might want to study in the model to decide on criticality versus non-criticality. Again, this is just a sort of a cartoon example of a theoretical chromatography step, for example. So this is just to conceptualize it, that you may have several MABs, say, for example, and it just picks some process parameters that, say, flow rate might have always been critical in your platform, low ratio, just for example, might have always been non-critical. So you could show that it's likely that this would hold again for the next one in the series. But also, along that vein, maybe there are some process parameters that for some products were critical, but some were not critical, so it's not quite set in the platform that some are always critical and some are not. So you might need some product-specific studies for these. So the idea here is just to kind of represent that you can have a combination of prior knowledge and product-specific data in the dossier, and you can justify what to study and what you can omit in your studies based on previous submissions. And I guess moving on from that, so that's just deciding on criticality. Okay, we use prior knowledge as a tool to help us decide what's critical and what's non-critical. But could it then be used to justify the ranges, or for every single product do you need to study in small scale every single range for every single process parameter to justify the edges of the ranges? And I discussed this a little bit earlier, that the way I see it, it's a system that's got inputs and outputs. The inputs are your process parameters that have certain ranges. Your outputs, if it was a chromatography step, it might be size exclusion, might be charge yield, or so on, whatever it might be. So you could build a model that shows that for very similar products, the inputs are always in this range, and we test the outputs, and the outputs are always within this certain range. So we know that when we run it within a certain range, we're always going to have a product of acceptable quality. So I think there is some scope for building a model here based on prior knowledge of previous submissions. And it could be from small-scale studies, full-scale process validation data, and ongoing process verification, because, of course, an awful lot of knowledge is accumulated subsequent to approval, which can be drawn on. So for each combination of the priors of the inputs, there are these known outputs, which can be consistent across products, if it can be shown. So you could develop a model theoretically for each unit operation to show that those ranges, or justify to us that those ranges will always result in acceptable quality. And then possibly that might be easier for the non-critical ones, and then maybe for the critical ones it might have to be justified that the model holds by some obviously product-specific data from the critical ones. Of course, this is just an idea. I mean, these are avenues you could explore, but this approach has not been used before. For any type of approach like this would benefit from scientific advice, because it's not an agreed regulatory principle yet. These are just ideas for this workshop that we can, for food for discussion later on. And as I keep coming back to, in terms of combining product-specific small-scale studies with prior knowledge, the way I see it, prior knowledge can be used to complement some gaps in process validation data or small-scale studies. So you have your product-specific data. Then obviously you have some gap in knowledge because you haven't studied it for your actual product. That is covered by prior knowledge. But it's how to explain this in the dossier. That could be tricky, but I think it's a very important issue. And just thinking of it conceptually in terms of the process validation requirements, and this is particularly relevant for accelerated approvals, where traditionally you have a lot of small-scale studies, so you can reduce the extent of large-scale process validation data. Prior knowledge now can add to this to reduce the burden of full-scale process validation data. And then for those accelerated approvals, which you'll hear later on, you could have a combination of prior knowledge, small-scale, and concurrent validation to build an overall control strategy or knowledge base about how you can assure quality of the product. And again, just looking at it sort of conceptually, where you have a combination of small-scale, full-scale, and prior knowledge. You may have studied some in previous products in small, some in full-scale. You may have data from prior knowledge. And there are ways of combining this because, again, it's often a combination of inputs and outputs. And one way which you could convince, I guess, the regulators, and I think, again, this is particularly relevant for accelerated pathways, is because prior knowledge is used to plug a certain knowledge gap because you haven't studied it for your particular product, you could file with, I just call this an enhanced control program, IPC program, whatever you want to call it. So you agree that we don't have all the data from our product specific of our product, so we're using some small scale or some prior knowledge to plug that gap. And to reduce the risk, we will file additional controls. Now, as regulators, we know you don't want those controls throughout your product lifecycle because it's a lot of tests and extra expense and so on. So you could file a post-approval change management protocol where we agree with the regulators and industry up front, okay, you've got these additional controls to justify the maybe slightly increased risk or lack of knowledge. But those controls can be removed post-approval once you accumulate the additional knowledge when the product is approved and you have additional batches at full scale and so on. So there's a way of agreeing up front. As industry probably don't like additional tests, but we can agree that you file them, but we'll remove them later on if you get the knowledge which was maybe covered by the prior knowledge if you get it at full scale. So, and here's just a cartoon to conceptualize again where prior knowledge can be used in process development. It's all the way along from the preliminary QTPP, defining the CQAs, defining the CPPs, the small-scale studies, and process validation. And how do you document it in the file? Well, for the CQAs, what's the similarity with other products? That's a discussion on how you show they're similar or relevant or so on. Defining the CPPs, what risk assessment and small-scale studies are applicable across products. For the small-scale models, was prior knowledge used to decide the outputs of the study, and are the manufacturing steps the same across products? So the models could be transferable or parts of the models. And setting the outer boundaries of the PARs, clearly describe what is product specific and what is prior knowledge in terms of justifying the ranges of PARs. And where does prior knowledge come from? So it would need to be just stated where it's actually coming from, is it from approved products for so on. Justify how it's relevant for the product rather than just go, here's some prior knowledge in another file, go look that up. It's how is that knowledge directly relevant for my product? What am I using that knowledge to show, and how is it relevant such that you can convince us that we don't need these additional product-specific studies? And what are you trying to show? What is it replacing in a way? That's very important. And discuss any remaining uncertainties. If you're using prior knowledge, then presumably there is some uncertainty because it's not a product-specific studies. So how will that uncertainty be reduced? And over time, particularly for accelerated development, this could be reduced in terms of protocols to agree upfront on what further data will be gathered post-approval, because, of course, the benefit-risk decision is slightly different for accelerated products, and we can agree that reducing some uncertainty and risk post-approval. So just some final thoughts. I think you can come up with a holistic approach, so a combination of prior knowledge, small-scale, full-scale, post-approval change management. There's scope to use it in risk assessment and assigning CPPs and setting the ranges of PARs. And we'd encourage you to challenge the regulators, because we do encourage submission, because this is all still quite theoretical, but we're happy to see it, and we would encourage you to use scientific advice to agree upfront your plans to use it. And it will require clear explanation in the dossier, so the easier you make it for us, the less questions you'll get. But I think finally, we're becoming very more closely aligned. I think you'll see some of the ideas in this are very closely reflected in the case studies later on, so I don't think we're that far apart. And I just have a quote there, which I quite like, is that people are more comfortable with old solutions than with finding with old problems than new solutions. So I think we do need to come up with new solutions, and this is the forum to do that. Another quote I like is that if you've ever tried, ever failed, no matter, try again, fail better. We won't get it right today, and we may not get it right in the first submission. So we're learning, I guess, as you're learning. So we do encourage submissions. It's not all our problems aren't going to be fixed today, but I think over time, as we see more prior knowledge, we'll become more comfortable with it and used to it. And if we repeat this form in 5 years, I think it'll be a very different place. There you go. Just the next, that's the end of my talk. So we have coming up next, Ogilvie from Pfizer, who will have the general considerations, and we have 3 case studies that will come from Amgen on prior knowledge and defining process parameters. Frank Zettl from Roche, who will talk about prior knowledge and process validation. Anne-Marie Murphy and Nancy Cauwenberghs from Eli Lilly and MSD will talk about prior knowledge and viral safety and resin lifetime studies. So we may leave the questions till the end rather than after each session, because I think it's quite a packed session, so we'll just discuss all the issues at the end. So I'll turn over to Ron now. Thank you.
Thank you, Seán. It's always a pleasure to follow an Irish fellow talking about failing better. I'll try and fail better. It's interesting. This was a could you just give a presentation about how small molecule development in manufacturing is supported by the use of prior knowledge. Could you just? It's a kind of a tricky task, but that's what development scientists face all the time. We're always delivering accelerated, tricky tasks. And we try and find ways to make that task as simple as possible to deliver. So what I'm going to do for you is give you a flavour of the kind of scenery you'll pass when you're on that development science express train that takes you through the development track. We've heard a lot this morning about platforms. I would also argue that some of the track in front of that train is really important too. And the better known that is to you, maybe the faster and easier your program can run. I'm speaking this morning really on behalf of EFPIA, so there's a number of colleagues who've contributed to this set of slides and story, and I hope I do it justice, but we'll see how we get on. That's the slide there I'm looking for. Right, so you're on this development express train, and let's think a little bit about platforms. Because, the platforms we've heard about this morning are things that are closely related members of a family. And no one, I think, has many concerns that you might share something very close with a close related member of your family. But interestingly, when you get into small molecules, at least for drug substance, things seem much more diverse. So is it possible to apply prior knowledge to drug substance small molecules in a platform sense? And how does the term platform apply in drug product manufacturing and development? Now, Matt touched on some of that with a simple pediatric example this morning. But actually, because you've got such a body of scientific understanding that we base our industry upon, this kind of prior knowledge utilization is happening all the time. So even for diverse drug substance development, you're working from a basis of a real understanding of the fundamental science, chemical processes, kinetics, thermodynamics. You understand from your manufacturing experience lots about manufacturing, chemical engineering, manufacturing operations. So it's interesting that this use of prior knowledge is happening all the time in the industry, and this is not a new workshop in that sense. It's not a new workshop for regulators either to hear about prior knowledge being used in risk assessment or scientific advice. The interesting thing is that, with it being happening all the time, what's not happening all the time is it's changing the dossier content. And it's kind of interesting to think why you can have platform and well understood tracks, but it doesn't necessarily change the dossier content. And I think really the future ahead of us on this track is where it impacts the dossier content you see. Someone else showed a picture about the types of knowledge that's in play here. Now, there's definitely fundamental, publicly available knowledge that we're all trained in as scientists. That's that general scientific knowledge. And certainly in your submissions, present and past, you've seen a lot of the stuff at the bottom, which is data from specific product design and selection. You've seen a lot of that specific information. But interestingly, that tranche of knowledge in the middle, which is that commonly understood contextual knowledge from the industry, from a company who's made similar products in the past, you haven't necessarily seen that used either often to change the dossier content or maybe in an optimal way. And it could be that that's the most valuable part of the knowledge that we really start to need to play with as we're trying to optimize the processes going forwards. It's interesting that that kind of bucket, whilst it's most valuable, that contextual knowledge, it's not necessarily the easiest stuff to share. What goes in a dossier? For example, Mark's lovely presentation about vaccines and generalized Q-tips and generalized platform design spaces, he's not going to publish that because AbbVie know what they're doing. They don't want me to know what they're doing. But interestingly, the regulators are inside the tent. So that contextual knowledge around that middle valuable piece is something that all companies, in theory, would want to share with regulators. As long, of course, as it doesn't complicate the review process. As Seán says, no point doing something if it's going to get in the way of your approval or get you far too many questions. So we need to make it work for everybody. Working for everybody. This is the famous industry slide that says, let's do less. No, it's not. It's the slide that says, if industry can understand things better, use their prior knowledge more effectively, then actually by doing more of that, you get some advantages. Now, we've had discussions around the validation and how does we validate how many batches we need. That's one very simple, pragmatic part of the story. However, it's a lot broader than that, because if you invest in using prior knowledge in an optimized manner, you can also change development speed. You put yourself on a faster track. You can change if you like, the outcome from your development program because you get better robustness. You get less chance of failure through ununderstood risks. You also get to a position where this prior knowledge, when well applied, can support you future innovation. It can help you manage change in different ways. But only, of course, if you can share it effectively with your regulators around you. So I think it's really interesting to see how the context around this matters for all parties. And actually, there's some things that are in play now in the industry that not everybody necessarily shares. Because there's almost a lowest common denominator of information that goes in the CTD. Now, actually, that's not necessarily optimum for any party or any stakeholder. Drug substance diverse. Lots of different drug substances. Therefore, of course, no prior knowledge can be applicable, right? No, it's not like that. If you think about design being knowing how to achieve what you need to achieve, then in fact drug substance is always designed to do that. There's a massive amount of almost taken for granted ability of the industry to make drug substances of huge complexity and diversity because of the fundamentals of chemistry and chemical engineering. Massive knowledge there. But interestingly, it goes beyond that, because industry can design in the right isolation points, the right purge points, the right conditions such that impurities are minimized. So actually this design from fundamental principles affects those real outputs in a very prior knowledge-based way. even though the structures are different, the prior knowledge use is there. To share some of that, how do you share the purge of an impurity? Well, industry are collaborating just now through LASA to have a consortium approach to sharing that kind of information in a database such that all companies are going to be using it in similar ways and presenting it from a database that the regulators can use and interrogate. So that's one way to begin to share consortium knowledge. But there was drug substance which is diverse, however, is still well controlled through the consideration of prior knowledge. What about drug product? Let's start at the bottom of this slide. The validation piece. We've heard this morning that when you start to understand your process through the application of prior knowledge, it can impact upon potentially the validation expectation at the point of submission. That's already available to us in the European system and guidances. So industry really happy. Regulators have understood and given value because industry knows how to do the same thing over and over. Because validation is just doing the same thing over and over. And you're happy that prior knowledge can impact upon that. We're all happy. But interestingly, when you get into drug product process development, there's a much wider set of things that can come into play. You've got all the platform understanding for the manufacturing processes, your understanding of excipients, your understanding of equipment, scale up, etc., that allows you not only to do the same thing over and over, but to do things right first time. When my next train comes along and gets ready to run down the track, I've got a good idea of what I need to do to make it successfully reach the next station. So actually prior knowledge can help you do the things you need to do in a right first time way. Interestingly, it can also help you build in processes of the future. A continuous manufacturing process is something that handles a process such that it gives you the quality you need all the time. So not only are you doing it over and over and right first time, you're doing it all the time, under control. So in fact, the future looks more and more like platforms, because platforms and continuous manufacturing approaches are not dissimilar in their overall philosophy, if I can use that word. So we know that we can do the same things over and over. We can see ways to apply prior knowledge to do the right things first time round and continuously. And we can also use prior knowledge to do things differently. We can understand how we can manage a development program and scale it up, either by using continuous manufacturing, so scale up is not really a requirement anymore, you've changed that paradigm, or by understanding the chemical engineering aspects of scale-up so that you can predict them, model them, and, if you like, predict the final result of a large-scale experiment. That, after all, is all that validation is. It's a large-scale experiment to predict. So scale-up can be done differently. Site transfers, because they use the same kind of engineering science, can in principle be done differently when you start to bring in that prior knowledge of a company's experience. Company A that's developed many, many products like this, you'd expect them to scale it up pretty effectively. Company Z, which is coming to that kind of product for the first time, it's going to take a lot of thought on their part and all our parts to make sure that that product is as easily scalable and transferable. So it can do that kind of development change differently with prior knowledge, but it can also allow you to change your inputs. It can do that whilst maintaining the outputs, as Seán said this morning. And this business about management of change, life cycle management of change, becomes a fascinating part. Someone's already said PACMPs are part of using prior knowledge. That's exactly right. They predict how you get the outcome you need. They're mostly provided to you on a product-specific basis. But actually, as some people have said in the audience already, Jean-Louis and others, maybe the value of PACMPs is when you see how broadly they can be applied by bringing the product understanding not just to one product, but to a family of suitably appropriate products. that kind of generalization of our understanding and our regulatory response to the understanding sounds very difficult and dangerous, but actually it's been done already in our regulatory system for some very complicated things. If you think about a BCS-based biowaiver that drops you a bioequivalence study for a site or product or manufacturing change, in fact, what you're doing is putting it in the context of prior knowledge, so it's that you understand how to manage that risk in a different way. So actually, prior knowledge is all about understanding and approaching risks and dossier content in different ways. It could be about modeling. It could be more about prediction. It could be certainly a little bit different in terms of how we use generic risk assessment. And it's not just for validation purposes. It's for process development. It's for process scale-up, transfer, control understanding, the design and validation and change of analytical methodologies. All these things can be linked back to prior understanding in a way that industry can optimize how they present that to you. We've heard the simple examples of packaging changes. That is great to do that. But actually the whole science of stability and prediction of stability and understanding that from extrapolation and prior knowledge, the future is there in front of us now in this kind of train journey we're on. I think we're here today because the optimum use of this platform and process prior knowledge is actually beneficial to everybody. It's got to have a win for everybody. So it's got to make industry more efficient, more rapid. Hopefully patients get higher, robust products quicker. The regulatory process is simpler. The change management process is simpler. So in fact, applicability of prior knowledge to the whole industry and the whole of our regulatory system is a great thing to do. So I know that we're worried about you need to justify it. Of course that's the case. But actually that justification isn't as difficult perhaps as we think it is because you're inside the tent. It doesn't all have to be public knowledge, it just needs to be shared in appropriate ways. So I should stop rambling. I think my last point would be, I said we were on a train. Well, you know, the next part of your train journey is not listening to the safety warning that says, mind the gap, you can't do that, mind the gap. It's actually, we've delivered where we need to be. The next station is where we need to be because we're using our scientific understanding in different ways. So I think on EFPIA's behalf, we thank you all for the opportunity to come and talk about this. But to give you the depths of all these different tendrils of prior knowledge, difficult. But to keep you entertained for 15 minutes, hopefully I failed better.
So now we turn over to Bob Kuhn from Amgen, who's going to talk about prior knowledge and process parameter definition.
Okay. So mine's going to zero in really on the process parameter definition using platform data. So for process parameter definition, you basically need to understand and assign the criticality of the process parameter and identify CPPs based on their impact to CQAs, and then the ranges that you can operate within that deliver acceptable performance for those CQAs. And an effective process parameter definition really requires an effective risk and knowledge based framework, and I use the word inclusive knowledge-based framework. And my talk today is really going to focus on using platform data as part of that knowledge framework, because there can be, as we talked before, a strong correlation in unit operation of the platform of the effective input parameters on CQA outputs. So most companies use some sort of sorting tool when we're developing the control strategy, where at some point we look at what we know about our process parameters. And this is a 10,000-foot view of our sorting tool, where we basically look at the severity of impact of a process parameter on the CQA and the frequency of occurrence. So basically, things that have a high severity of impact to CQA, or maybe a lower severity but higher occurrence, are sorted into the potential CPP pool, where we require more in-depth knowledge, often product-specific knowledge, to assess their actual criticality and justify the ranges. And then there are lower risk parameters which we identify as non-CPPs based on previous knowledge. I'll say previous knowledge that don't require additional studies, and then we need to document that justification. So previous knowledge, I said instead of prior knowledge, because that knowledge can come from early development, it can come from manufacture for that product, it can come from general scientific principles, and it can come from analysis of platform data. So the effectiveness of this sorting really is contingent on the overall knowledge base. So I put this slide just to kind of, because it's germane to what I'm talking about. The use of prior and platform knowledge is really dependent on what the knowledge is used for. And we can ask different questions. We can ask qualitative questions. Does this input affect the output of this unit operation? And that's kind of qualitative knowledge used strategically. And it's quite different than using knowledge quantitatively to predict the exact outcome of a unit operation. And the expectation for the exact similarity of molecules, the exact similarity of the processes, may be less for qualitative understanding of the effect of an input on the relative output. So we can really use, in a justified fashion, prior knowledge based on how it's used and the associated risks. So one of the things we've been doing is formal prior knowledge assessments, and these are almost like experiments based on our platform data where we ask a specific question, does this parameter impact this output for this unit operations, and we'll ask that for a number of different things. And the materials for this are the not prior knowledge sources, and we'll look at the relevance of it relative to the quality of the studies, relative to the similarity of product and processes. And then we have methods for analyzing this data, often taking a lot of quantitative data and making it kind of a qualitative output. And I'll talk about one example of that in a minute. And then we'll basically review this, analyze the information, draw conclusions, draw recommendations, and develop reports which then can be used by the teams to reference for their risk assessment and process development. So here's just one example. We have different examples. This one was actually published 5 years ago of an example where we can take quantitative data and get qualitative risk assessment impact out of it. So basically, this is just a scoring thing where we look at perturbation magnitude of the process parameter, how far outside of normal operating range relative to the effect on CQAs, no effect to large effect. And obviously, if something has a large effect on a CQA over limited range of process parameter variation, that's going to be high risk. If it has no effect over a large range of process parameter perturbation, it's going to be low risk, and there's various degrees in between. This just is one example of where we did this on the, I guess the y-axis is different reports where we looked at the output of various operating parameters, process parameters on a CQA. You can identify there's certain parameters that come up as red a number of times, or yellow. So we can look at this just qualitative. Whoops, I need to introduce this. So I have a very short case study where we apply this to a cation exchange chromatography, which is one of the options in our platform process, but it's used for multiple molecules, binding to loop mode. Its primary purpose is to clear a few impurities. And so we systematically evaluated data from 14 monoclonal antibody products. We have a lot more now, as well as looking at extensive manufacturing data for these products. So this is just an output, and this is a radial plot just showing negative or positive impact on two different CQAs, and they're so similar. I realized I made a mistake when I was reporting the slides that I put the same plot twice, but they're very similar output, and it makes sense because it's the same mechanism of action: charge-based separation, differential absorption, p-cutting, and the same process parameters had a major effect: the amount of product loaded, the elution conditions, and the stop collect where we're excluding impurities. So we also looked at a lot of manufacturing data, and this is basically the amount of impurities for two different impurities in the load versus what comes out in the pool. We look at this amongst many different products. It's just a few example graphs where we show that a strong trend in the amount of product impurity loaded translates to a high level in the pool, and this also informs our strategy. So I'm trying to make up time, so this is my last slide. So this is just an illustration of how we use all this knowledge to really have a focused PC strategy, process characterization strategy on the impact of the step on quality. So our prior knowledge assessment for this operation, unit operation showed really clearly identified what the high-risk parameters were, and also identified parameters which were very low risk that were non-CPPs. I didn't show this data, but process parameter interaction shown in multiple DOE models were not practically significant for this step. I didn't show this data as well. Raw materials were a significant impact of the step. And as I showed a minute ago, we really highlighted that the feed stream quality or the amount of impurities loaded impacts the performance of the step. And for other impurities, there was, we showed significant excess clearance across multiple products. And this really informed a focused, effective, quality-minded risk approach to the process characterization strategy, where we really focused our experimental studies on a small number of potentially critical parameters to assess their final criticality and the acceptable ranges. And then it highlighted the importance of feed challenge or spiking studies, where we challenge with higher level impurities to assess the clearance capability of that step, the performance requirements for the previous step, what the previous step is required to deliver, and then the output of the required output of the step, so that it informs the control strategy and the testing requirements downstream. So that's basically my example.
Okay, thanks, Bob. So now we're going to go to Frank Zettl from Roche, who's going to talk about validation efficiencies from QBD and prior knowledge.
Okay, thank you for the opportunity to talk here. Also, thank you to the industry associations here for the trust that I can give a meaningful contribution here. And hello world, I learned that this is broadcasted live.
Massive audience.
So I want to jump right into an example where I think we have a success story of how we applied QBD principles and prior knowledge. So this is a product that we launched a few years ago. It's Gazyvaro. And in this case, we already used the prior knowledge to understand, to design the molecule. So we have a pretty good structure-function relationship in many cases based on the prior knowledge. In this case, this molecule was designed with a glycoengineering approach in the FC region. So this led to an increased alpha-acetylation of this molecule. It's an IgG1 with effector function. And this alpha-acetylation increased the ADCC, the antibody-dependent cytotoxicity, by around 100-fold compared to the standard of care, as you can see on the upper right panel. So based on this, we already knew, of course, that the alpha-acetylation is an important CQA that we need to control and to study in the process validation study, and that happened, and that was kind of one important CQA, but of course not the only one that we studied. But I think it's a nice illustration of that it already works somehow pretty well. So prior knowledge is used to reduce risk. I think it's clear to everybody that we cannot study all process parameters that you can think of, even if we are using highly effective or efficient methods like DOE, design of experiment approaches. We still need to select parameters, and we still need to decide to which rigor we investigate parameters. Do we need to identify interactions or not? So with our QBD tools and this systematic procedures, we do guide the experimental effort. We filter out some CQAs that do not need to be studied again. We have maybe a lot of data in a lot of cases. We do out-filter parameters that don't need to be studied again. And maybe we even do filter out complete studies for unit operations. I think prior knowledge helps, of course, in the judgment of the severity, but mainly it drives down uncertainty to get this risk score and to kind of decide to study the right things. I think in many cases we do it right. Right. Okay. I think the challenge we have, and probably in many big companies, we already have the challenge to kind of have a summary of the prior knowledge that we have internally. I mean, Roche is a big company with many places separated in many places in the world. So what we did here, we compared the affinity chromatography steps, and this is the case study here I want to make from 11 monoclonal antibodies. And each process was individually optimized, so that was no two same processes there. But of course, we identified commonalities across the different maps. So when we did this, we looked at these studies. We saw similar low risk and high risk parameters, and this is true across different map formats and also across different affinity resins. So the aggregation of this prior knowledge is now then used to support the further risk assessments that we are currently doing and that we will do in the future. So this is an impression of how it looks like. So you can see a lot of green. So these process parameters have never impacted, and there's a whole area around there. The CQA in charge here, so what we call here impurity A, but there are also clusters where you see that, for example, process parameter process parameter 2, has always impacted, or in many cases has impacted the impurity A, and then there is kind of different rigors of the assessment here. I mean, we have identified CPPs. It's shown in red. We have sometimes identified minor effects, that's shown in blue here, where we have seen a detectable impact, but maybe that did not lead to a CPP in this case. And then we had low impact CPPs, that's depicted here in yellow. But what you also see, and that's maybe a challenge that we can discuss later, is that, for example, in MAP A and MAP 2, which are probably pretty old products, these parameters probably were not studied. So what does it mean if we file something like this for us? I think that poses really kind of questions with regard to change control, and I think that's something I would really like to discuss. Okay, so now this is, I would say, pretty well accepted and a common approach in the industry, what I would like to do now, I would like to move forward. So we are continuing on our QBD journey and also the development strategy and the modeling that is for us, I think, a very important topic for the future. I think machine learning will enable really an enhanced data-driven knowledge. What we have seen so far is kind of an aggregation of knowledge in reports, but machine learning and all these modeling techniques will enable a data-driven, even more data-driven knowledge. I'm not saying that the other thing is not data-driven, but this is, really going down to raw data somehow. I think the usage of computational models will be applied throughout the life cycle of a product, and the computational models and the methods behind this will change over time. We will leverage automation for knowledge building studies, and when I talk about automation, I think it's both. It's kind of robotics, so kind of experimental automation, but it can also be the automation of the data evaluation, which probably will progress pretty fast. So in essence, we will use the available data and the mechanistic understanding to create these computational models. And we want to apply this for new products, so where we have maybe a limited data set, and we want to support the analysis of this with based on these limited data sets. So I think in essence we will get really better and more product knowledge using less resources here. A case study I have here is also related to the protein A chromatography and the reuse studies. So this is something where we have to say that this is really a very feedstock consuming and time consuming effort with, personally I would say, limited value. We anyway do a verification at manufacturing scale. We have seen really in I guess all the products that no impact on the CQA was detected so far in the resin lifetime for protein A. So we really want to not study it again and again. We have seen, on the other hand, that yield might be impacted. That is important for us. And we have now developed a modeling approach. This includes a series of 2 partial least square assessments or evaluations. And this will predict the chromatography performance with regard to yield. So basically, we have small-scale data. We use the chromatograms of these small-scale data, different information in the chromatograms, and then we get this what is called a multivariate batch evolution model. What you can see on the right side here. So this is not a chromatogram anymore. This is a multivariate representation of certain phases of the chromatography. And you can see also here, maybe it's too small, that there is already information. The blue ones are the early cycles and the red ones are the late cycles, that there is information in there that we can model. So basically what we are aiming here is we want to in this case, the model was generated using small-scale data from MAP-A. This model was then further trained and extended on small scale with MAP-A data, but also we used large scale data for MAP-B, and we really trained this to predict the impact of reuse cycles on yield. So we have seen patterns across the data sets that are predictive for the impact of the reuse cycles and yield, as you have seen before. And on the right-hand side, you can see the gray dots are the modeled data, and the red crosses are the actual data, that this works pretty well. I think the challenge that we have here, this is still a limited data set, so we want to perform here a cross-validation approach. So that means I think we can avoid overfitting here, but we want to make best use of all the data we have. So the data of MAP-B that we are kind of that's what we want to predict. They are also used to train the model, but we apply a cross-validation approach. And I think we are probably in good shape that the models are capable to replace the current small-scale reuse studies and that we can kind of overcome the studies. We would be able to monitor the predictive patterns during the manufacturing, and we would aim to continuously improve these models with new data from manufacturing. So the data that go into this model will change over time, and also the model themselves can change over time. And I think this is probably also challenging for the regulators, I assume. But this is hopefully a discussion we also could have. Maybe not everything today, but okay. And I think I'm done with this. Probably I saved another few minutes, not sure.
Thanks, Frank. And so our last case study will be from Marie Murphy from Eli Lilly and Nancy Cauwenberghs from MSD. We're going to present on using prior knowledge to streamline viral safety and resin lifetime studies.
Okay. Thank you for the opportunity to speak today, particularly in relation to bringing viral safety and viral clearance as a topic into the prior knowledge discussions. And this case study is presented from a monoclonal antibody perspective and also from a vaccine case study perspective today. So both Nancy and I will present this case study. Common to both monoclonal antibodies and the vaccine case studies is the utilisation of platform-based processing for viral clearance and viral safety. And that's really helped to streamline process development, ensure we have robust ranges for manufacturing, and also to ensure orthogonality for viral clearance. And that's common to both case studies in this instance. So for the monoclonal antibody-specific case study, I'm presenting this today on behalf of a viral safety consortium from EBE. I'm representing the EBE team. Okay, and two specific components of viral safety that are in scope for discussion today. The first one relates to virus nanofiltration. The second relates to affinity capture chromatography. But to begin with nanofiltration, there is quite a significant amount of published data, both from a regulatory perspective and also from industry knowledge, as to the types of viruses that are typically used to challenge small virus filtration, both for early phase or IND applications, CTA applications, and also for late phase marketing authorizations as well. And what's common to all is we see time and time again the robustness of these small virus or parvovirus retaining filters in terms of their robustness to clear viruses of all different sizes. And each and every time we relate the fact that the small viruses in particular, so the parvoviruses in particular, are considered to be worst case contamination model virus to be removed based on this size exclusion mechanism of action. Yet early phase and commercial applications will typically continue to include log reduction factor claims for both large, intermediate and small viruses. So as part of our prior knowledge, or an example of prior knowledge here, is how to better utilise our viral clearance studies and complete those, for example, using the worst case small model viruses as our worst case virus, and therefore then extrapolate back this type of log reduction factors and extrapolate those back indeed to the larger viruses in lieu of product-specific large virus clearance studies. We know that this has been successful already and has already been demonstrated for investigational medicinal products. And in terms of reporting this in the dossier, the achieved log reduction values for the worst case studies for the larger viruses will be interpolated from the smaller ones, even though they, in theory, could achieve higher safety factors. Okay, so the consortium, obviously there's a lot of literature that's available in the industry is previously published, and there's actually new data coming online all of the time just to indicate just how robust these small or parvovirus retentive filters are for clearance of different virus types. And what the consortium has done in this instance is provide an evaluation of our own products across 5 different small virus filter brands. 31 products, for example, in this instance were evaluated. And again, what we see is the log reduction factors and the robustness of retention for larger viruses in comparison to smaller viruses is reiterated again here within this consortium set of data, therefore reiterating the fact that small viruses are indeed a worst case challenge. In addition to the model viruses that are utilised to challenge the virus filters, there's obviously significant emphasis on understanding and qualifying the worst case conditions that are utilised for virus filtration under GMP conditions, and thus ensuring what the potential critical process parameters are. And one of those potential critical process parameters which has evolved over in more recent history, has been the potential impact of process interruption as a parameter that must be taken into consideration, particularly for its potential to affect filter efficacy. And this schematic here is previously published information, which just indicates some of the differences with respect to small virus retention if the filters are challenged with multiple stop-start sequences, for example. And so what we are understanding now is that depending on the filter type and the operating conditions that are utilised, filters can may be either sensitive or insensitive to process interruptions. So as part of process knowledge, either internal process knowledge but also platform data, we're able to understand and therefore propose on the basis of this prior knowledge to obviate the requirement to perform product-specific process interruption studies once there's a thorough knowledge of the insensitivity of that virus filter to such interruptions, and that has been assured. Moving from virus filtration into the second unit operation that we wanted to briefly discuss is back to affinity capture chromatography, or protein A-mediated chromatography. The mechanism of action here is obviously different. The viruses flow uninhibited through the column, followed by wash sequences, whilst the product of interest preferentially binds. The ICHQ-5A guidance that is obviously is utilised for viral clearance studies can be interpolated to indicate that each product should be measured for virus reduction following resin multicycling, in addition to evaluations in naive resin. But prior knowledge, both published data as well as industry and in-house knowledge, would indicate that affinity capture mediated clearance is not directly impacted by typical multicycling, especially if the manufacturing process utilises conventional cleaning regimens and it's operated under GMP conditions. And as a consequence of this, one additional proposal here for prior knowledge would be that with the use of robust process monitoring, particularly for other more pertinent performance-related attributes such as antibody, basically binding capacity, but also antibody breakthrough, one can more appropriately analyse the potential for resin performance over time, or degradation of resin performance over time, in lieu of having to measure log reduction values for each of the specific model viruses on multi-cycled resin on a product-specific basis. Again, the Viral Clearance Consortium assessed the available published literature that's available here on multi-cycled resin for viral clearance, and again looked internally to our own particular products, which have included in this instance monoclonal antibodies, fusion proteins, and also plasma products that are cleared using affinity capture resins that include base stable and pore glass resin types. And what we have in this diagram here is just an indication of the differential between aged resin and naive resin and its capacity to clear the different model viruses that are indicated. And again, in the mainstay, what we are seeing is very, very little differential with respect to capacity to clear these model viruses, whether the resin is naive or whether it's aged anything between 50 to 300 cycles. Okay, so to summarise, the EB Consortium Group obviously acknowledged the importance of appropriate viral safety and how this must be ensured for our patients. But we're calling specifically for feedback on these two proposals for these two unit operations to determine whether this prior knowledge would be appropriate to utilise in these cases to obviate requirements for product-specific studies on this basis. And as part of this workshop, to best determine what those minimum expectations for utilisation of this prior knowledge would be, so that we can continue to further our process development. So I'm just going to hand over to Nancy on the vaccine case study.
Yes, thank you, Marie. So Nancy Cauwenberghs from MSD. So in the next few slides, we actually want to illustrate that we believe that there are also opportunities to streamline resin lifetime studies for vaccines. And actually in these slides we will illustrate how we have started to evaluate the use of prior knowledge to support resin reuse across vaccine serotypes. So the case study concerns actually a multivalent vaccine. And similarly as for monoclonal antibodies, so chromatography is really a step that is utilized for the purification of many of our drug substances. However, today we typically have resins that are dedicated to each serotype specifically, and they have a limited number of uses. So this, similarly as for monoclonal antibodies, results in a discard of costly resins, and there are certainly opportunities to optimize our manufacturing facilities. So for the multivalent vaccine, for which we have started to do this evaluation, we actually have a wealth of prior knowledge because this vaccine is already on the market since many, many years. And so this prior knowledge comes from developmental studies. So we have shown that the physicochemical properties of the serotypes are similar. We have also information on the manufacturing process unit operations, which are nearly identical for all serotypes. And we actually also have the luxury that we benefit from a small-scale model that we have already utilized for several process changes and that has been shown to be predictive of commercial manufacturing. So since this product is already on the market since many years, we have also commercial manufacturing history data, in-depth knowledge of CPP, CQA. We have all the wealth of continued process verification data, and also that clearance of process and product-related impurities have been shown to be similar across serotypes. And so exactly what we have started to do is to use these process stream data, which are commercial data, but serotype specific, to evaluate the qualitative and quantitative characteristics for the clearance of impurities. So you see here actually 4 graphs, but these are representative of the different serotypes that are contained in the vaccine. And so for the different impurities, we have started to analyze those impurity profiles based on statistical criteria to analyze the comparability. And so based on that, actually, we will be evaluating the serotypes for which we believe we can recycle the resins. So actually the next step is based on these data from the individual serotypes. We will do this selection, and then we want to confirm the feasibility of this resin reuse across serotypes in our small-scale model, which I said, which is actually predictive of commercial manufacturing, which is, of course, of a great advantage to us. So then ultimately our proposal would be to submit and gain regulatory approval based on our prior knowledge, further supplement that with the data from the small-scale model that demonstrates that we can reuse resins across serotypes, but still it will be for the same vaccine, so the same final product, and then monitor that further the performance at scale under the PQS system. So overall, we believe that also we've increased knowledge from individual serotypes, so process product knowledge, small scale and historical commercial scale, that resin reuse across serotypes within a specific polyvalent vaccine and for extended uses may be justified. Thank you.
Okay, thanks, Nancy. So we're just running a little bit behind, but we still have 20 minutes or so for discussion. So I guess we've got AB of some particular questions that they would like us to address, but not to. We can discuss both the EB questions and also other discussions on general on the session. And just before I open the floor, it's just to note that, I mean, in those slides we see from Amgen gave us data on 14 MABs, Roche on 11, EB consortium on 31 products. So that just goes to show what could be submitted in a dossier in terms of the amount of prior knowledge that's there. You know, it could be, if it's science driven, it could be quite convincing if you have data from so many products to justify it. It's not just you coming with 1 or 2 previous ones. This is years of experience. So it's just, and I thought it was interesting to note that prior knowledge is available. So I guess you do have those particular questions. We have a slide, 2 slides there, which just kind of highlight those. So I don't know, maybe Johannes might like to address some of those, considering he's our viral expert.
Of course, I'm Johannes Blümel from Paul Ehrlich. I will be happy to give feedback on the 3 topics mentioned by Marie Murphy. So the first was using a parvovirus only for the nanofiltration as a kind of worst case model. And as you said, this is a practice which has become, this practice has become also widely used already for clinical trial substation. And I suggested on my own this years ago at PDA meeting that it would be an option. Yes. So I think for filters where there is this experience that the size is the main driving factor, we have accepted this because these viruses which are going to be replaced are really much larger. So the reovirus or retrovirus, so there's no concern. What we have been discussing also amongst assessors is, of course, this touches a little bit the principles outlined in the ICHQ-5A guideline. Just to quote a little bit, it says where only a rodent retrovirus has been found, process-specific evaluation using specific model viruses such as murine leukemia virus should be performed. So some of my assessors colleagues read this quite strictly, and I could here see really colleagues adhering very strictly to that, and so this might for me an issue which should be or could where there would be some reflection to needed to give also some regulatory clarification. Yes, when thinking of that as one question which also comes into my mind, and where I'm happy is if we rephrase such things, or if it's clarified what we would expect also for new filters coming on the type. So I would be careful in saying it can be always so. So we have to be very careful for new filters, because the principle is not only size exclusion, it's also so the thickness of the filters and other things, and all the flow dynamics. And the second point was the low pressure and pressure release, and also here we experienced a really, I think, increased understanding of the mechanism, because we experienced that of course the pressure release might have an impact. If the pressure is released, the viral particles might diffuse back out of pores and find new pores where they can go through. So from a general scientific understanding for that, it's a critical process parameter and kind of worst case to the low pressure. We are even discussing whether it's the pressure release or whether there shouldn't be any low minimum pressure, and we have seen few dossiers, or not all dossiers, where there's any specification of low pressure. That's why we have started to ask on this parameter. Of course, this effect might differ depending on the filter manufacturers. However, I have to say I'm not yet convinced by the data that the publication that you presented. It was from 1 month. It was not using the parvovirus, a slightly larger virus, and I'm not sure whether these conditions or these effects are seen in the same way under each specific manufacturing conditions. We clearly understand that filtration is not cheap, it's very expensive, it's a cost driving factor, and that there's always also a need to push as much as possible product over the filter. So I'm a little bit afraid that under certain specific conditions we could see effects that we did not see under this prior knowledge. So here the recommendation would be to accumulate more knowledge. There is one manufacturer even clearly saying that it is a worst case conditions and so on. And for the others, I think there's still some work to be done. Last comment on the reuse of the protein A column. Also here we have, of course, long experience and an increased scientific understanding what's going on, and the picture that we have in the moment, and this has already been published, is that the virus might co-bind with the antibody and co-elute. So, and we have seen this trend when the protein R loses its capacity to bind and purify the antibody, the more virus goes also in the flow through. So from this understanding that there is a mechanistic idea behind arguing that the reuse, if the protein A is destroyed, this is not a worst case for the virus co-purification. So this might also be something where we would be open for discussion. I looked in the ICHQ5A, and as you said, quoting again, it's quite flexible worded. Some estimate of the stability of the viral clearance after several use may provide support for repeated use of such columns. And of course this then again comes to discussions also amongst assessor, and maybe here some further clarification may be needed. Thank you. And yes, last word on the protein A, we have even seen situation, of course, all has to be considered in a context where we did not request any validation of the protein A if you have other robust clearance steps, as you showed in your meeting. And your last question, there's a platform to continue to such dialogues. Of course, we have here some opinion. I think this has really grown over 10 years by performing specific virus clearance meeting with industry every 2 years. Thank you.
Okay, so we open the floor to questions. Mats?
No, maybe a comment on the vaccine case study here, that as it appears here, and again that depending on we're talking particularly that these different, if they are pneumococcal or they are the polysaccharide vaccine, they may be of slightly different chemical nature, but it appears that this will be checked in the small scale model that an acidic, neutral, or basic polysaccharide will be captured, and as such, I think the way here would be to have a kind of concomitant validation with this, and I would say under certain circumstances that could be acceptable, which would then need, you would have to state some kind of acceptance criteria for the performance for that, for each and every run, and also justify what would be rejection criteria, when it's not usable any further. But if you can do that, I think you can build a case with these data. You are verifying in each and every commercial run, and for that reason maybe it gives us higher assurance than if you, as today we are looking for the small scale data, and then you do it all, you have your concomitant large scale verification, but without sort of insight. So I think that should be a possibility, and maybe invite others. But in the same manner, I think that the proposal for the MAB could be doable as well. That if you have the model that looks really predictive, you have your in your large scale data, you have each run has some acceptance criteria that it needs to fulfill, and you have justified your acceptance criteria. That is also, at least I think, as a basis could be a doable thing, and not really asking for hundreds of runs in small scale before submitting the data. So, but leave to others. This is my personal view on this.
Stefan.
Thank you. Steffen Groß, Paul Institute. Just two remarks. One thing is automation is really a good thing that you mention automation, not only for the process, so the process parameters, but also for data evaluation. And there's a famous term, batch record by exception. I guess we are not there yet. I guess this means you have only to review the batch record again if you have a deviation. I guess we will not accept this that you only review a certain batch record if you have failures or deviations there. I guess this is a nice idea for the future, but we are not there yet. And the second thing follows up to the validation issue. I like this validation, the discussion, having, let's say, the small-scale studies substantiated by additional prior knowledge data. But to be honest, I hear always we have everything under control, even at large scale. But to be honest, we have really very rarely seen submissions where all 3 consecutive batches were really successful. So we have many, let's say, submissions where we have 4, 5, 6, 7 batches because some of these batches failed. So it seems that not everything is under control. So we have seen some other data here. So you should be really careful to, let's say, leave out everything. Thank you.
Matt?
So it's not about the viral clearance, but I think it's linked actually quite closely. Seán, you talked at the start around an approach where you'd, I think you said, take a model based on prior knowledge and test that model in your new product. So your prior knowledge from other products would be testing your new product. So I think I'm just a comment on the terminology there, and to reflect how I'm hearing what you said to check that it's right. I think what you're talking about is your process understanding comes from a previous product. And what you do for the new product is you verify that model. So you're looking at the extremes of the input materials, the extremes of the process that you've identified based on your product understanding, and there's a verification approach. And that verification approach requires very much less experimentation than it would a de novo approach where you develop that model for your product. You're simply taking it from one to the other. So you're nodding. I just wanted to check that was what you were referring to.
Yeah, I mean, that's an idea I put forward, and it would have to be obviously agreed. But the idea is that because you are substituting in a way some of the product-specific knowledge with your prior knowledge, it is a model that any model has to be validated, but it could be validated post-approval. And this could be agreed in a protocol up front. And in terms of what data will be gathered in the future in the product life cycle to justify that prior knowledge is indeed applicable and does successfully underpin the control strategy going forward. And as talking to Matt's point, part of that verification protocol could include additional acceptance criteria and so on until such time as it's very clear that the prior knowledge is directly applicable and there's no risk of unintended consequences or so on.
It's a very helpful concept. Like just if I can just reflect on thinking about it, I mean it's obvious that the long-term provision of additional data is part of your thinking, and that makes perfect sense. But it's interesting for me. It's a slightly different approach from how we've often talked about verification in the past, whereby for a specific product you build a small-scale model and then you do verification at the full scale. Here we're talking about the model effectively comes from the prior knowledge. There's limited verification done for that product, and therefore, if it wasn't for the prior knowledge, it would be seen that there was potentially gaps if it was a brand new product without that prior knowledge. So I think that's a very interesting proposal, and one I think that a lot of companies will be interested in considering how to explore that.
Andrew, had a question?
Yeah, Andrew Leonard, Amgen. Seán, if I understand correctly, you're saying about filling in knowledge gaps using prior knowledge, but then also adding additional controls to compensate for that. Well, isn't it, in fact, isn't it the opposite of what we actually do, in that we use our prior knowledge to determine the severity of an attribute and determine whether it's a CQA? It seemed to be the other way round, because then we determine our control strategy according to the risk associated with that criticality.
I think that idea is more leaning towards accelerated pathways, where you have a very limited development time, so the amount of product-specific data you have could be quite limited in some instances. And there, the prior knowledge may be filling quite a larger knowledge gap. And for us as regulators, there is some risk there. And one of the ways to reduce that risk is to have some additional uncertainty post-approval, which ties in a general sense with sort of accelerated pathways in terms of there's a benefit-risk balance. Knowledge accumulates over product life cycle. We have slightly less knowledge at the time of approval, but the knowledge continues to accumulate, and that's maybe even the same on the clinical side and as well. So by moving the bar forward, where you have less data, I guess, at submission, and we agree up front to accumulate more knowledge, there's some additional. You could use some additional controls to justify that you're using an awful lot of prior knowledge to justify your control strategy.
So it's more of you'd apply a more risk-based approach to it, and it'll depend on the attribute and the case.
I think it's all a risk-based approach, and it's all linked to the benefit risk. And I know Keith mentioned earlier on in passing, it depends on the type of application. And indeed, for certain, say, accelerated applications, it's by definition an unmet patient need. It's a very serious condition, so there we can accept more risk. Maybe some of the risk is a lack of knowledge of product-specific data, which is plugged by prior knowledge. So the risk is always in our mind, and also the benefit risk in terms of the indication.
Just to add a little bit on that, I think it's never black and white. I think what we should admit, so to say, and what we've always done, is that any application, any development plan consists of a combination of prior knowledge and new own product-specific data. And part of what you're doing during development is always driven by prior knowledge and platform knowledge and scientific literature. And the whole package should be acceptable at the end of the day, and should form a consistent justification of what you're doing. And that's also related indeed to for example, accelerated pathways and benefit risk, but also much closer to home, I think. If you're having a fairly straightforward manufacturing process with a CHO cell line and a monoclonal antibody, then and you're not claiming a very large parse or a design space, your whole package will look different, and the use of combination of prior knowledge and product-specific data will look different if you compare to if you want a design space for a completely new kind of protein which we have never seen. So the point being, I think it's not black and white. Any application will contain a mixture of product-specific and prior knowledge data, and the whole package taken together should provide sufficient assurance, whatever sufficient assurance in a specific context is, of product quality.
Brian?
We're just coming towards the end of time, so I just wanted to bring it back to these questions that we were said we'd come back to during the day. So based on what you were talking about in your presentation, it seems like the use of prior knowledge informing risk assessments is one of the key places where we could see its value, defining CQAs, CPPs, etc. And I suppose there seems to be, from the discussions, the regulators are open to looking at the data that's been presented in the case studies and considering it. The next question is that assessment has to happen in a formal way, so in a regulatory procedure, in a scientific advice, or in a pre-submission meeting, or in a dossier. So can we think about it? In one of the slides there was a suggestion that the viral safety information could go into the appendix of the 3.2a appendix in module 3. Does that seem reasonable, that type of information will go in there? Likewise, in Bob's example, he talks about the risk assessments of 11 different monoclonal antibodies. Where, Bob, would you intend to put that in the dossier? How would you intend to present that information? And from the regulator's point of view, would that approach be reasonable? I think if we could have another minute or two on this before we break for lunch, it would be helpful. And Nana?
Nana Kruse from the Danish Medicine Agency. I think Matt already touched upon this, and I would, from a regulatory point of view or as a regulator, not, well, I will find it anyway, but I would not prefer the option of having it located in module 3A, the appendices. I would like to see it where it's used, so that when you try to convince me that a certain process parameter is not critical, I can see from previous data or whatever located in that area of the dossier that why you think you can use the same data or whatever. So as Matt say, they are kind of replacing some studies, or are provided instead of some product specific. You can rely on previous generated data. I would like to see it where it's replacing something, so to speak, or replacing data.
Yeah, I mean, I'd fully support Nana on that. I mean, I think it's an easier proposal to go, here's our product-specific studies, here's what we haven't done, this is covered by prior knowledge, and this is why we think that prior knowledge is applicable and covers the extra risk of not having that data. It's more challenging to go, here's what we've done, for the rest of it, please see elsewhere. You know, that's very challenging. I think it does need to be integrated in the dossier in terms of what is product specific, what is prior knowledge, and how those two facets come together to ensure that there's appropriate control and so on, and the process is well designed. But by cross-referring to other places, it makes it a little bit challenging. And, you know, as we kind of keep saying, try and make it easier for the regulators. It makes your life easier in the end. But, you know, it's not an issue that's decided from a regulatory point of view, it must go here or it must go there. These are things I think are still up for discussion. As Brian says, we're open to seeing it in regulatory submissions, and that is where we really, you know, be challenged with it and have the discussions on what's the best approach.